lecture that's already posted for you for the census. The ones that, come after are, the lecture regarding taste and smell. This is at the end of that. So taste, smell, vision, hearing, all of those are our equilibrium. All of those are our special senses, where all of those receptors for those special senses are found in the head. Okay, and so now we're gonna just jump to our general senses. And our general senses cover touch, pressure, temperature, sensing temperature, pain, sensing what's happening with our muscles as far as stretch of our muscles, vibration. And so these general senses have what are called simple receptors that are out in the peripheral nervous system. And these simple receptors are actually the ends of these sensory neurons. Their dendrites have been modified and made into receptors to pick up whatever it is their job is to sense. Okay? So now again, we're talking about general senses that cover our tactile sensations, touch, pressure, vibration, but then also temperature, also pain, also proprioception, being able to figure out where your body is and or different parts of your body is in space, right? I can know my hand is over here. And that there's movement, okay? We're moving as well. And also being able to feel really light movement on my skin because my hairs have been deflected maybe by an ant crawling on the skin and so forth. So thinking about our general senses, and let's think about the ones that are either non-encapsulated or encapsulated. Meaning encapsulated, they're kind of wrapped in some connective tissue. Non-encapsulated receptors for the general senses include our free nerve endings. And then on our practical list, let's look at that while we're, the list of the skin, free nerve endings on there. Our tactile disc on here is called the, there's two different things. So Meissner's corpuscle, that's gonna be something else. But for us, we have the tactile, the Merkel's disc. We'll come back to the Meissner's. And then we have our hair follicle receptor, so that's here on our list is the root hair plexus. And then we'll see those other ones as well. Okay. Here, the encapsulated, they're wrapped in some connective tissue. There's where we'll list the Meissner's corpuscle that's on our list. The Pacinian corpuscle, it's on our list, and all interchangeable with lamellated, meaning concentric circles. Okay, that corpuscle, the Pacinian, then the Ruffini corpuscle is on here, interchangeable with Moelis corpuscle. And now these are things that we should be able to find on the model. Again, the skinnier one there, I believe we can only find 2 of these things on that guy, but on that one, we should be able to find all the rest that are on our practical list, okay? So we're gonna have to be able to, so I would take pictures of that box one, but actually you have a picture of it in your lab. There's a picture of it according, and it's, the key is there as well, yeah.
Dr. Colston, what's the other name for the root hair plexus again?
Hair follicle receptors.
Oh, thank you.
So root hair plexus is interchangeable with, the lecture hair follicle receptors is referring to that. On our lab practical list is being called the root hair plexus. Okay. All right, what else is on our list here? For lecture purposes, we have this Krauss on here, on the lab as well, the bulb Krauss. And then we're gonna look at the muscle spindle and the Golgi tendon organ that correspond to our muscles and our tendons not overstretching and tearing. Okay, let's cover our first non-encapsulated simple receptor. The three nerve endings of sensory receptors. These are unmyelinated. These nerves, these neurons these nerves are unmyelinated all over the place. Okay? We have a lot in our epithelium. They're responding to temperature, pressure, and pain. Three nerve endings. Very abundant in our epithelium, across the dermis and the epidermis. So at their ends, at their distal ends, at the ends of their dendrites, you get these knob-like structures, okay? And those are the receptors. And they are picking up pain, temperature, pressure, itch. So here are the three nerve endings. Three nerve endings. See if you can find that on your model according to the key. This is not our model, unfortunately. This is a great model. But doesn't look too different than our model. So let's go to our key. Do we see the skin block? Three nerve endings, anything that says that? Here. You absolutely. See if you see free nerve endings on that. When you see those, it's perfect with numbers on it.
So I know what it is. Okay, what I'm confused about is specific part, the tree, or is it the entire tree? I'm not sure.
Keep that one there.
Like, is it the neck of the tree, or is it the tree? It's not in the, It doesn't say that. It says Merkel's. This is Merkel's corpuscles.
Okay,
Can you
so
ask
this
the
is
question?
free nerve ending on the
It's
box
a tree.
one, right?
The branch of the tree.
I don't think it'll be a corpus, so.
I think it's incorrect here. I don't think it's an entropy. It's not an entropy.
What number is that?
48.
Is that what's on there? Oh.
It says
Oh, I
it's
see.
corpuscle.
Merkel's. What is that? Supposed to be Merkel's.
This guy is genius. But is it the entire tree or just the? You can ask him. I'm guessing it's just the.
So this big? No. Yeah. Like that.
Oh, she said the.
Get more like that.
The what? Pink? I don't know anymore. I don't get
I don't see any of that labeled on this picture of the box model. Unless it's. Right. So. So yeah, I don't see free nerve endings on that model. But nonetheless, we are going to remember that their job in the skin is to pick up pain, pain and pressure, some itch. Okay. And then I'll look over that again to see if I see anything, but so far I don't see anything on that model, unfortunately. The Merkel's tactile disc. Do we see that listed on our model, in our key? lab purposes, Merkel's is not on there, so don't worry about this one, but this one, because we find this is stragglazolin, as that's what it is. It has this, Merkel cell that's in, kind of connected to this, large nerve. The Merkel's disc is responding to light touch and light pressure. Okay, so here are Merkel's discs. Start staining cells in the epidermis, like there's the basal stratum basale, here's a Merkel's disc. And again, they're usually associated with a nerve. Here's the nerve, and then here's the Merkel's disc associated with nerve. Again, this is a great model that shows all that stuff, but ours does not, unless it's talking about, and it's not on our list, so we don't need to worry about that. But for lecture purposes, we do not, okay? But for identifying in lab, you just need to know those six, being able to find those 6 things on the list under the skin, okay? The hair follicle receptor is that plexus, okay? The root hair plexus. So we have a nerve plexus that surrounds the hair follicle. And so anytime the hair gets deflected on our skin, that'll send a signal that there's something on our skin causing that movement. Okay, so this is gonna respond to hair deflection, like blood flowing on us, or light touch. And so this, the job of this receptor is to monitor any distortions in the hair on our skin and any movements on the surface of our body. Okay, so those we should be able to find. Everybody see the number for that? See, hair nerve, 39. Find it on a picture. And then find it should be associated with a hair follicle. Remember what that looks like on the model? Histologically, here is the hair follicle receptor. Also called the root hair plexus. Another root hair plexus, and another. And on your model, it should be something associated with the root, hair root. You see something there at the hair root.
I see it now. So that's what it is.
Now for some encapsulated receptors. These are nerve endings that are gonna be encapsulated in some connective tissue. And these are responding to different types of general senses. Again, general senses, touch, pain, itch, vibration. The Meissner's corpuscle. That fella's on our list, right? Let's try to find him on the model. So let's go to your key, find Meissner's corpuscle, and see if we can find him on that model. And again, take a picture of the model. So you have that, I would take better pictures. This one is not the best picture in my opinion. I would take my own pictures. Okay, of that box model. We only have 2 in the room. Meissner's corpuscle is gonna be in those, remember the dermal papilla? The heels of epidermis that push up the epidermis. So find those on the skin model and, okay, so I'm gonna, since we have 2 on this side, we're gonna all have, I'm sorry, I only have 2. And then we're gonna have to give 1 to this side of the room, okay? And then you pick which side of the room you wanna, and so that everyone can get some of that model. Okay, so the jobs of these Meissner's corpuscles is discriminative fine touch. Discriminative touch means I can look and I can feel something and I know what I'm touching, without even looking. Okay? And they detect light pressure, low frequency vibrations.
Make sure you don't put tongs.
Or like you can't feel yourself scratching the itch. What is that? Is that like the nerve?
You can't feel yourself scratching it.
Yeah, like if you
Can't-
you feel yourself
can't satisfy the itch.
Yeah, but the itch still.
I don't know what that is, but I will say that itch is a big research field. There are people who devote their lives to studying itching. But yeah, so I don't know what that is if you can't satisfy an itch. Okay? And it is a big field. Itch. Okay. So here we are, so we're looking in the dermal papilla, the heels of the dermis that push up into the epidermis, and we'll see these Meissner's corpuscles. We can find them on our slides. They're kind of tough to find because they look so much like the dermis that they're hard to really distinguish. So it takes a good eye to find them, but they can look a tad different than epidermis. A little bit different. Okay, the Meissner's tactile corpuscle. Did you find it on this model? Looks like that. Our model is red. You know what number corresponds, or?
It says 44.
44, which unfortunately the model is not numbered, so. But we have the key, on the key is number 44, and so that red, found it? I think so, look on the key, and the number is 44, see what that's pointing to, and find it on the Pacinian corpuscle. This is gonna be found in the dermis, but we also find them in other places, like the tendons. Here we're thinking about, there is lamellar, meaning consisting of concentric circles. Okay? And this guy's job is to pick up deep pressure and high frequency vibrations. So these are the receptors that pick up earthquake. When we start feeling the earthquake, they're picking up this vibration. Okay? Yeah. So the Pacinian corpuscle looks like this histologically, concentric circles. Pacinian.
Oh,
And so then, we see that listed on our model. No. Oh, yeah, it's 20, it might be 25. The city. Corpuscle, 25. See, where's that? 25 down there, pointing to.
looking like a little watermelon.
But concentric circles, right? Perfect. Okay, and then the Ruffini's, we found that? No, the Pacinian, found that? The Ruffini's corpuscle, also called the organ of Ruffini. This is sensitive to deep, continuous pressure and stretch. And let's see, Ruffini, do we see that listed?
Brushes.
Hmm.
Is it brushes?
What number?
24.
Oh, yeah, that's what we got. If it says Ruffini, then that's what we're gonna say, yes. And what's he look like? Yeah. Mhm. So kind of looks like this structurally. Kind of reminds me of an Aladdin's lamp. And on the skin model, does it take that shape? What number you say again? 24? Okay. Yeah.
Yeah.
Okay. Alright. The bulb of Kraus is picking up cold temperatures and some light touch. Okay, so we'll have these in our mucous membrane. Okay. So let's see. See anything? Kraus's corpuscle, number 22 on the box. So see what is pointing to? The little red one. And one of them is sliced open. So this one has Sicinian, or, the Sicinian is here. This is supposed to be free. Can I keep my attention? Yeah, it's not listed. Yeah, so it's not, I would call that the free nerve ending, but the model does not. So I don't see free nerve endings listed on the key, and I don't think it's on this one either.
Not this one, but the other one?
Do you have the city of corpuscle?
No, it's look, you can have the white one. The white one.
Oh, so this one
The Meisner's corpuscle, number 1. Where is that? Okay, so. Yep. All right, so on this skinny one, the narrow model, we can see Pacinian corpuscles and Meisner's corpuscles. The key is in the integument map. Okay, but, so like here are Pacinian corpuscles on this model, and here are Meisner's corpuscles. Okay, so here are Pacinian corpuscles and Meisner's. That's the only thing that we find here. So far, on neither one of them do I see anything that's a free nerve ending. Okay? But one thing that we can use both of these for is the hair root plexus. Okay? Even if we don't see nerve endings at the roots, on this one we do. Okay? On this one we do. But even over on that one, you can be asked what general receptor is associated with this structure. And if it's the hair root, then you know it's the hair root plexus. Okay, even though it's not pictured, you can be asked what receptor is associated with it. Yeah.
So why isn't this one root plexus? I mean,
I don't know, I did not make the Trust me, I would have made the model would have been much better. I didn't make this model, I don't know. You gotta call those folks and ask them, how come that is? All right. Okay, so we saw the bowl of Kraus. Okay, so let's look at some others that are not on our practical list, but things we still need to know as lecturers who learn. The muscle spindles, these are muscles found in the bellies of our muscles, and these are responding to stretch so that we don't overstretch a muscle. So it's gonna pick up as a muscle is lengthening, okay? And then it also provides some, letting us know where we are in space, different body parts, okay? I can tell my hand is over here. Okay, so a muscle spindle is always gonna be embedded in muscle tissue, so you'll always see some muscle cells around. Okay, so if I was to ask about this, I would show a picture. So please be familiar with these pictures in the model or in the lecture, okay? This is how, if I was gonna ask you anything visual, it would be a picture, okay? From the lecture, because they aren't on our models. there's another muscle spindle embedded in between the muscle fibers, muscle cells. Okay? There's a cross section through muscle. Okay, so here's the muscle fibers, and here is a muscle spindle embedded within the muscle.
What are those inside it? Are those, like the little
I
rings?
don't know what those are saying. Okay? Muscle spindle. Yeah, I don't know if those are, yeah. Okay. So, a muscle spindle, again, is gonna be embedded within the muscle belly itself, picking up stretch and being able to, respond to the stretch so that we don't overstretch a muscle and tear it, okay? And then we have at the tendon that anchors a muscle to a bone, we have another receptor called the Golgi tendon organ. The Golgi tendon organ is picking up stretch in the tendon. We don't wanna overstretch our tendons and tear those. So we have a receptor there that's picking up stretch of the tendon. The Golgi tendon organ, in its name, right, tendon, picking up stretch of the tendon. And so these, Golgi tendon organ are part of a reflex. So as our tendon is stretching, okay, that Golgi tendon organ will pick up the stretch. And then we'll get, if we're starting to overstretch, we'll get a reflex to cause relaxation of that muscle. The body's saying, oh no, you're gonna rip this tendon, so we're gonna relax that muscle, automatic, okay, as a reflex, before we tear the tendon. Okay, so here is tendon, and here's a Golgi tendon organ. There's muscle, so that muscle spindle is someplace in there, picking up stretch of the muscle. There's a Golgi tendon organ, and this is, muscle, this is Golgi tendon organ. Okay, so I just wanted to finish that up for us. That's all of our senses. We're gonna head into the heart together. So get a bottle.